Okay, so can you hear me? Can you hear me? Can you hear me? Okay, so can you hear me? Okay, so can you hear me? Right, so let's start with today's lecture or today's session basically. So in today's session we have a lot of stuff to do. What we have to do, we have to do basically four PGRs, plant growth regulators, and we are supposed to do four types of movement. Right, today's class is going to be a very short class, around 40 minutes or 45 minutes. Be with me, be patient, and we will finish it off in no time, right? Just tell me, can you hear me or not? Okay, nice. So let's start with this discussion. So how, okay, so today is first of all, don't write anything. This was from Oxine. So now today's discussion is going to be what? Today we will start off with gibberellin, gibberellic acid, Ga. Right? Can you see on the board? Gibberellic acid. Now, what will you do? You will do the discovery first of all. Whenever we are going to talk about a plant growth regulator, we will discuss it in 5-6 points. So the first point comes is the discovery. Now, in discovery, you have to write who discovered the hormone, when he or she discovered the hormone, and how the hormone was discovered. Whether it was an accidental approach or it was a guided approach, whatever it is. Like auxin discovery was an accident and all that stuff. And the story, you have to basically know the story, what happened actually. So this you're supposed to do on your own. Now we are coming to the chemical nature. Now in chemical nature, what you are going to do? Now chemical nature, gibberellin. Let's talk, right? Do let me know. Do comment in between whether you are with me or not. Otherwise, it becomes very difficult to understand, right? Now coming to the chemical nature. Now gibberellin is acidic in nature, right? It's acidic in nature. It's acidic in nature. It is termed as gibberellic acid. Gibberellic acid. Okay, gibberellic acid. Right, and they are denoted as Ga1, Ga2, Ga3, and so many. There are almost 100 Gas present. Okay, sorry, I wrote 1000. It is supposed to be 100. So there are more than 100 Gas are there reported. Now out of these, Ga3 is most studied one, and one of the first to be discovered. One of the first to be discovered. This is important. Ga3, so gibberellin is acidic in nature, and Ga3 is one of the most studied gibberellins and one of the first discovered gibberellins. Right. Now another thing that you have to keep in mind, that gibberellins are also, they are acidic, but their chemical nature proper is they are terpenes. Right. Now what are terpenes? If you have seen any painter, or if you have seen painting work going on in your home, they use a oil known as turpentine oil. Now the turpentine oil is mixed with paint works, mainly oil paint works, and they are used to remove the paint from your hand. So turpentine oil and these terpenes are basically same chemically. Now gibberellic acids are basically terpenes from chemical structural point of view, and their acidity or basicity point of view, they are acidic, and that's why they are termed as gibberellic acids. There are 3 types of gibberellic acid: GA1, GA2, GA3. I mean, more than 100 there are, but out of them GA3 is the most studied. Right. Now we are coming to the biological function and use. Now before that, let's talk about the occurrence of gibberellic acid first. Right. Let's finish off the discussion of occurrence. Are you with me? Now, occurrence of gibberellic acid, if we talk about, gibberellic acid is not found in root, not found, or it has no role in root. Gibberellin has no effect on root. No effect on root. No effect on roots. This line is very, very, very important. Sorry. This line is very, very important. No effect on roots. Right. Now, this was the occurrence. Now we are left with what? We are left with the biological function and the use of gibberellin. Now gibberellin is a plant growth promoter, PGP. So what it will do? It will do all this stuff that we mentioned over here. Plant growth promoter function. Now let me clean up the board a bit over here. Right. Can you identify this part, the plant growth regulator part that we were discussing the previous day? Yes or no? Now over here we will discuss things. What biofan going on right now? I'm not understanding what you're trying to say basically. Now over here in plant growth promoters, what we are supposed to know, they will promote growth and they will not promote abscission, they will not promote senescence. Right. Now in case of growth, there are two characteristics of growth: increase in size and increase in number of individuals. Those are the twin characteristics of growth. On the other hand, on the basis of observation of growth, I did not understand what you're trying to say. I mean, biological functions. Yeah, biological functions are going on. Okay, so fun. I mean, what kind of fun it can be? Plant hormones is one of the most boring chapters, so it cannot be fun. Yeah, biological functions. Biological functions, and then we will do uses. So basically, understand one thing. Now, biological functions are manipulated by human beings as uses, right? So first of all, if you don't know what are the biological functions are, you cannot understand how they are used. So whenever we do biological functions and uses, first of all we will do the functions, then we will come to the uses. I explained the biological function and uses stuff, the DNA example, right, in the previous class, isn't it? In the previous session I described that with the DNA cutting example, molecular scissors and all, right? Now over here, on the basis of observations, we have two kinds of growth. We have microscopic growth and we have macroscopic growth. Now microscopically, what gibberellin does? Gibberellin does cell elongation. So microscopically it does what? It does cell elongation. Not division, but cell elongation. Right. Okay. That means cell elongation falls under which category? Increase in size, isn't it? Cell division falls in the category of increase in number. After cell division, you will get two daughter cells. But after cell elongation, what will you get? You will get increase in the size of the cell, right? So basically, under the twin characteristics of growth, cell elongation falls under the size part, right? Now we will come to the macroscopic features of growth, that is observations through naked eye. Now first we will come to the shoot. Now in shoot, gibberellin does something known as bolting. Bolting means what? Let's understand what bolting is. Okay. Now for that, let's move to another place on the board. Yeah, nice. Now if you observe a plant stem properly, now plant stem has some parts known as nodes, now swollen parts in the stem. This is the land, below that it's the root, the soil level, and this part is the stem. Then the stem, what happens, there are certain swollen parts on the plant stem, and from these swollen parts branches arise. Understand? Now these swollen parts from which branches arise are known as nodes. Right? Now if these are the two nodes, then the region between two nodes known as internode. Isn't it? Place between two nodes. Right. So by means of cell elongation, what gibberellin actually does, gibberellin causes internodal elongation of stem. Internodal elongation of stem. Right. It causes internodal elongation of stem. Sorry, actually I'm not able to stick to the schedule of taking classes on Mondays and Thursdays. Right now some of my college online classes are going on and they are continuing till late in the evening, around 7:30. So sorry for not able to stick to the schedule. Okay, so what gibberellin does, gibberellin causes internodal elongation of stem by means of what? Cell elongation. Gibberellin means elongation. So that means what will happen to a plant under gibberellin influence? Suppose these are the node intervals. Now what will happen? Now the node intervals will increase. Can you see the node interval over here, and can you see the node interval over here? So basically node interval increases, right? The node interval increases. So that's a function of gibberellin, and this increase in stem height is known as bolting. I mean this internodal elongation. So basically the terminology increase in stem height through, yeah, stem will elongate, ultimately elongate. Yeah. And see, there are two kinds of elongation of stem. Stem elongation can take place in two ways. Suppose this is my stem, over here we have three nodes. Now one stem elongates and over here we have four nodes. Okay, this is a kind of elongation. And other elongation can be, over here we had three nodes, now over here we will also have three nodes, but the internodes will increase over here, right? Now you can keep the distance between the two nodes. That means you can conserve the internodal distance and you can keep on adding nodes. By that you can elongate a stem. Or what you can do, you can keep the number of nodes constant and you can increase the distance between the two nodes. That's how also you elongate a stem. So what gibberellin promotes internodal elongation of stem. And this increase in stem height or stem elongation through internodal stem elongation, this process is known as bolting. Very important. Write in your copy, bolting. Right. So what gibberellin does? Gibberellin does bolting. Now why you require bolting? So for that, let's take another example. There are certain leafy plants like spinach, like lettuce. There are plants which are grown for their leaves, like the leafy vegetables that we eat. The leaves, the plants whose leaves we eat, they are leafy plants. Now understand one thing. Suppose this is a stem. Over here we have two nodes, and from nodes there will be stems coming out, and from on the stems there will be leaves, on the stems there will be leaves. Right? Now if the nodes are very close, that means internodes are very small, then the leaves will get crowded, they will have bushy appearance. If the leaves get crowded or get congested in a place, then the chances of a leaf of getting good sunlight is reduced, because one leaf may get covered by the shadow of one other leaf. In that case effective photosynthesis will not take place. If effective photosynthesis does not take place, then what will happen? They will starve nutritionally, because glucose production will be hampered. If glucose production is hampered, they are nutritionally starved. Then what will happen? Their growth will get hindered, and if their growth is getting hindered, then your productivity of your field or if your farming will drop. So to increase the productivity of these leafy vegetables, what you do, you promote this bolting kind of action. Right? Understood? Understood? Right. Now basically bolting was the idea in case of shoots. So if we go back to our that skeleton, so shoot, they will be positive. Right? Now we come to branches. So now there's no such effect described for branches. Gibberellin has no described effect on branches, but as a plant growth promoter they will promote branching, but you're not, sir, you don't require to mention that, so I'm putting a cross. So don't mention branches in case of gibberellin. Now root gibberellin has no effect, so it will be again a cross. Now leaves, it promotes the growth of leaves. Okay, so for leaves it promotes the growth of leaves. And flower, now we are coming to again the idea of this BD segment. Now what happens in case of BD segment? Plant growth promoters help in achieving this puberty or flowering phase fast, so they quicken up the juvenile phase, isn't it? Plant growth promoters quicken up the juvenile phase, as we saw in the case of auxine, as we have seen in the case of auxine. Is the buffering gone now? Okay, so now are you with me? Okay, so now are you with me? Can you hear? Can you hear? Can you hear? Can you hear now? Okay, so let's continue with our session that we were doing. Right, where I left. Sorry for the small technical glitch. I had a internet issue. Where I left, just tell me where I left. Just do let me know where I left. Flower and fruit discussion to begin. Yeah, I was talking about the BD segment, and in that BD segment, how juvenile phase, the rate of growth in the juvenile phase is increased by plant growth promoters, right? That I told you, isn't it? All the plant growth promoters increase the growth. Yeah. So what will happen? So if that happens, if juvenile phase growth increases, then what will happen? Flowering will increase and fruit production will increase, isn't it? Flowering means puberty for plants basically. So if growth is enhanced, then flowering will be promoted, and if flowering is promoted, fruit is basically what? After the sexual reproduction is done, whatever is left is the fruit. So if sexual maturity is attained, then they will do sexual reproduction, and if they do sexual reproduction, then what will happen? They will produce fruit. So flowering will be positive, it promotes flowering, it promotes fruit formation, right? That means it promotes fruiting, but it does not promote fruit ripening, because fruit ripening means what? Ripening is your senescence, and they are not supposed to promote senescence, right? They delay ripening. Understood or not? Okay, nice. Now, so what? Now one thing that you are supposed to know, that is your uses. With this we finish off with our biological function. Now you are supposed to know the uses. Now if we learn about uses, what we have, first of all, the first use will be fruit growth. They will promote fruit growth, right? They will delay ripening, promote fruit growth, delay ripening, quicker maturity. Are you writing these things down? Are you writing things down? Okay, so fruit growth will be promoted, delayed ripening will be there. Then third point will be quicker maturity. Maturity means what? When you are mature, you're matured when, biologically speaking, when you reach your reproductive completeness. But our parents don't consider us mature by then also. That's a whole different story of sadness. Let's put that aside for now. So quicker maturity. Then there's something known as malting in brewery industries. In brewery industries, one process is known as malting. Now, gibberellin increases malt yield in brewery industries. Let me write it down somewhere. Yeah. So gibberellin increases malting in brewery industries. Brewing industries, right? Basically, this is the fourth point. 1, 2, 3 we have discussed, right? Now this fifth point that you're supposed to know, increased sugarcane yield. Now what will happen to a sugarcane? What will happen to the sugarcane? Now the sugarcane, basically what we do, we cut the stems, we chew the stems, and we savor the fluid or the sap, sugar-filled sap of the sugarcane, right? We imbibe that. So basically that's how we eat sugarcanes, isn't it? So that's how we eat sugarcanes. And now if internodal growth is increased in stem elongation, then what will happen? Then what will happen? Then there will be, the sugarcane part is basically what? The internodal part that we eat or that we savor. So that amount of that will increase, so sugarcane yield will get increased. So basically this was the idea of gibberellin. Now we are moving on to the third category, that is cytokinins. Now how cytokinin was basically, again let's get back to the structure of our discussion, that is discovery, whom, when, how, that you will do on your own. Now what is the chemical nature of cytokinin? Let's understand. Let's understand the chemical nature of cytokinins. Right. Now cytokinins are modified nucleotides. They are modified nucleotides. They are known as modified nucleotides. Nucleotides I have already taught you, and one such cytokinin is known as kinetin, whose chemical name is N6-furfuryl. N6-furfuryl aminopurine. This name you have to remember, that kinetin, the name of kinetin is, you have to remember, that is N6-furfuryl aminopurine. Exactly, contains phosphate, not related to. Very good. It shows that you can recall that. So basically this is the chemical nature. Now occurrence. Occurrence of cytokinin. Cytokinin occurs where? They occur in three places. What are the three places of occurrence? Basically, three places are root apex, developing shoot buds, not shoot apex. Okay, so let me write down things properly over here for you. Root apex. Okay, now we have shoot apex, developing shoot buds, growing fruits. Now over here put a big cross on shoot apex. We are coming to the discussion of this, but just remember that cytokinin is present in root apex, in developing shoot buds and growing fruits, but not at all shoot apex. Right? Now let's come to the biological discussion. Now why this happens? Now what are the developing shoot buds? From these nodes, whatever comes out from these nodal regions, these are known as developing shoot apex. Okay, you with me? Right. Now what happens? That means if it is present in the root apex, it will cause root elongation. in the apex it is present, so apex it will cause elongation. Shoot apex it's not present, so it will not cause shoot elongation, it will not cause. Developing shoot buds, that means they will promote the branching of the root, that means it promotes branching. And in the fruits, so it will promote fruiting, right? So now let's come back to these discussions. Now if I say shoot, they promote branching. So basically shoot apex, whatever I wrote by shoot, basically that was shoot apex. They promote, but they promote basically branching, not shoot elongation. Can you understand this part? They promote root elongation, not a problem. Leaves, yes, they do promote the leaves growth. They do promote the growth of leaves. And flowers, obviously this is a plant growth promoter, so it will hasten off the juvenile phase growth. And flowering means what? Puberty reaching. That means it will promote flowering. And fruit growth we already talked about, it will promote fruit growth. So it will promote everything but not elongation of shoots, right? And now if we come to this microscopic discussion, then what will happen? In the microscopic discussion over here, the name is cytokine. Okay, so let me change over here, this part is deleted. Now the name is cytokine. That means it is promoting. Okay, so first of all, there can be, okay, in a plant, see on the top left corner, in a plant, this is the shoot apex, which promotes shoot elongation. And there are some growing shoot buds from which branches grow. Okay. Now cytokinins are not present in the shoot apex. So cytokinins are not present in the shoot apex. They are present in the developing buds. Over here they are present, right? And hence they promote branching. They don't promote shoot elongation, right? Understood? Okay, now cytokine in the microscopy, cytokinin comes from a word that is cytokinesis. Right. Now, microscopic work, what it will happen? Cytokinesis, that means cell division. Right? Right? So that means the microscopic part of its growth is cell division. Now quickly come to the uses of this. Okay, another thing that you are supposed to know is that it will inhibit senescence, it will inhibit your abscission, that's known. Now what auxin does, auxin promotes this growth, auxin inhibits this growth. That is known as apical dominance. That means auxin promotes elongation of shoot but inhibits branching of shoot. Whereas your cytokinin is doing what? It is cytokinin, if I talk about cytokinin is inhibiting elongation, it is promoting branching. And what auxin does was known as apical dominance. That means apex was dominating. But cytokinin does not allow that. And hence cytokinin is said to inhibit apical dominance or counteract apical dominance. Understood? Right. Basically, there's no such use of cytokinin. Basically, cytokinin is used to inhibit senescence, and cytokinin is used in research purpose in laboratories. Not much uses. Now we are coming to, first we will come to ethylene. Should I start ethylene? Apical dominance. Apical dominance. I'm writing it down. You have written this down in the previous class. Right, now we are starting off with ethylene. Ethylene. Right, ethylene. Now what happens in case of ethylene? Ethylene is a plant growth, it's not a plant growth promoter, it's a plant growth inhibitor. It's a plant growth inhibitor.